there are two books which are of critical importance, critical authority. And I want to add to that a third one. Let me express it this way. There is the Old Testament. And here we have just a page or a part of the great Isaiah scroll discovered at Qumran, which demonstrated that we have such a wonderful record, an accurate record of the Old Testament writings. And what Israel had to, the foundation of Israel's faith was her story. This is ancient Israel we're talking about. And I quote a couple of theologians, Hauerwas and Willimon, who say, in telling that story, in telling Israel's story, she comes to see herself as a people on a journey and adventure. It does not just happen that God's people tell stories. Story is a fundamental means of talking about and listening to God. The only human means available to us that is complex and engaging enough to make comprehensible what it means to be with God. So that's one story. The second story is a New Testament story, which is the story of Jesus Christ. And here is a page or a section of the Codex Sinaiticus with the monastery of St. Catherine at the foot of the Mount Sinai in the background, where Bible scholars discovered this amazing manuscript which attested to the accuracy of our New Testament. And Hauerwas and Willimon also talk about the meaning of New Testament story. They write, the early Christians did not begin with creedal speculation about the metaphysics of the incarnation. They began with stories about Jesus, about those whose lives got caught up in his life. By telling these stories, we come to see the coherence and significance of our lives as a gift, as something that must be told us. I was very impressed with the quotation from Monod that was given us by Jeff Talon this morning, who said, the only explanation capable of putting the soul at ease is that of a total history, which discloses the meaning of man by assigning him a necessary place in the wider scheme of things. So this is the biblical story. But there's a third one which is authoritative. If we are in fact the creation of God, if he is our author, if we exist because of his will, then the story, the history that defines us biologically must also have an authority. This might sound a little bit scary to people, perhaps, who are Christians. But the third story is a story written in our genes. And here's a page of text which describes some of that story. This is a middle-sized gene, which in fact describes one of my favorite genes, the P53 gene, which has a critical role to play in cancer biology. And I'm not a molecular biologist, but you can download this gene or just about any other, just sitting at your desk through PubMed, just a simple 3 or 4 clicks on your mouse and you have it. So, as a Christian, believing that our biology is God-breathed, it must be if we are the creation of God, then the story in our genes has an authority. It tells us something about our biology, something about our history, and so it describes something of what we are in the larger divinely given scheme of things. And a philosopher, Holt Ralston, has said the study of life, otherwise known as natural history, is in fact storied. We can read that story in our genes. The Earth's story becomes memorable, cumulative, and transmissible in genes. So although Jeff spoke about the language of mathematics, I don't think he can talk about the story of mathematics or the history of mathematics in the nature of maths itself. But biology is storied. And I just want to give you an example of the power of genetic history before I I move on and talk a little bit about the nuts and bolts of evolutionary history. And this is a paper which is to appear in PNAS, in the Proceedings of the National Academy of Sciences, in the next week or two. And it's a story of the mystery of what happened to the Romanovs. What happened to the last emperor of Russia and his family? And here we have Nicholas II and Empress Alexandra and their 4 daughters and dolls, and Crown Prince Alexei, who's sitting on the knee of the German Kaiser. And we know from history that they were murdered in the early days of the Bolshevik Revolution. And in fact, the remains of most of the family were discovered in 1991 in a graveyard, or an unmarked grave, should I say, near a place called Yekaterinburg. And here it is, marked by this arrow labeled number 1. But when people did the genetics on the bone fragments, they could only find evidence for 2 parents and three children, and a number of unrelated people who were servants and the court doctor. And so mythology grew up as to whether, in fact, the Crown Princess Anastasia and the Crown Prince Alexei, whose bones were not there, maybe they survived and they lived to tell another story. In 2007, a second grave was discovered nearby, indicated by arrow number 2 and the lower picture. And from this, DNA was from the bones exhumed, even though they had been burnt and treated with sulfuric acid, DNA was still extractable. And the remains of the 2 skeletons were linked in with the Romanov family. And we can see it here in this picture, where we've got part of the family history of the Empress and Tsar Nicholas and their 5 children. Mitochondria are little bodies in our cytoplasm which have their own little bit of DNA. And it has been found that there are 7 members, in fact, of this family in the 2 graves, linked together by their mitochondrial DNA sequences. The mitochondrion is about 16,000 letters long, those A's, T's, G's, and C's, which define the genetic story. And one of these stories, in fact, starts off with Queen Victoria, who was the grandmother of the Empress Alexandra. And all those blue circles I've drawn around different people, each symbol represents a person and each layer represents a generation, represent people with the unique Queen Victoria mitochondrial DNA story. They have the same DNA sequence. And so it is possible to tell from this that Queen Alexandra, that Empress Alexandra, is certainly a direct descendant of Queen Victoria, and that 3 of the children in the 1996 grave and the 2 skeletons discovered in the 2007 grave are also in that direct line, as indeed down here at the bottom is a surviving member of the family still alive, as indeed is this person over here in another branch of the family, as indeed is this male here. The square represents male, who is Prince Philip, the Duke of Edinburgh. So we have here a story told in DNA which relates a couple and their 5 children to Queen Victoria and her family. Well, mitochondria are passed through DNA, and the Tsar's mitochondria do not exist in his children, but rather they arise in, or we can follow them in the other side of the family, in the Tsar's mother, Dagmar, sorry, I've forgotten the word, I'm not too good on Russian, Maria Fedorovna. And there are, in fact, surviving members of that family here, that's an older study, and here, which indicates that unique mitochondrial sequence is still being passaged, and that in fact it does delineate, or it does help to identify the Tsar. Now there are other things too. We can certainly identify the Tsar from blood stains. This is a coat he wore from an assassination attempt in Japan in the beginning of the 19th century. And from those blood stains on his coat, the DNA can be extracted and related to fragments of DNA extractable from bones which had been buried in Russia for the last 80 odd years. So I think I've got too much stuff here, but we can just indicate from this recent history how our DNA tells a story. And changes in DNA can be followed from generation to generation. And so just briefly, I want to consider the story of changing genes. And here again is another example which comes from the literature of a year ago. We have different eye colours. Some of you have brown eyes, down here. Some of you have blue eyes, up here. And some of us have somewhat of a mixture because genetics is often very complex. Now, if you look in the DNA, there is a point somewhere in those 3 billion letters which define us as Homo sapiens, where there is one letter difference. And that is marked by the yellow in that little patch of DNA story. The blue on either side represents a point where a protein binds to that DNA to control a gene. And the middle of that protein binding site differs between the people in this room. Those of you who have brown eyes have an A in that site. And those of us who have blue eyes have a G. So what's the relationship between us? If you look at a whole lot of other mammals, you find that they all have A's. So it's the G's who are the mutants. So we are mutants, and also we are related. Because if you look at the surrounding DNA environment, it's very homogeneous. And that implies that the mutation which defines blue-eyed people arose something like 10,000 years ago in Central Asia. So blue-eyed people, we are cousins and bros, and we can trace our genetics back maybe just a few hundred generations. But there are other telltale signatures in our DNA which go back a lot further. And here is just a small part of the genetic story, something like 50 bases or so, 50 letters, which are in fact part of a gene which is implicated in one of the most common human genetic diseases, Gaucher's disease. It's part of a gene which encodes an enzyme called glucocerebrosidase. We've got 2 copies, and one of those copies has a 50-odd letter deletion. Interestingly, if you look at the DNA, at the genetic story of chimps, you find that they possess exactly the same deletion. And if you go further back and look at the genetic story, the genetic sequence, the genetic language of gorillas, they have the same deletion again. If you go further back and look at an orangutan, then you find that this gene copy is intact. They have the 51 missing base pairs, or 51 letters, and so does the squirrel monkey, as a representative of an anthropoid, a primate. Now, these are the sorts of changes in genetics which geneticists see in their labs all the time. And what it does mean is that here we have an essentially unique mutation, which is in fact shared by 3 species. And so the conclusion is ineluctable. It is overwhelming evidence that this unique mutation was in fact inherited by 3 species. humans, chimps and gorillas. And so I say this to set the stage for what is to come, that we have here very powerful evidence which Christians and other religious people must take very seriously, that the genetic story gives us a convincing argument that we are in fact an evolved species. Now here is a second story. All human beings have very high levels of a substance called uric acid in our blood. And this is because we can't make the enzyme called urate oxidase, which metabolizes uric acid. Why do we have high concentrations of uric acid? It's because in the middle of our gene that encodes uric acid, we have a mutation which has changed a C into a T. And a TGA triplet, a TGA word, encodes stop. These red circles represent the protein encoded by the genetic sequence. And whereas with crab-eating monkeys and rhesus macaques and baboons and owl monkeys, they've got the intact gene which encodes R for arginine. My cursor's not working. R for arginine. We in fact have a mutation, a particular mutation, which we share with 4 other species and which stops the protein. It truncates the protein. Now you wonder, surely this is a disadvantage to undergo the loss of a protein. One would think so. And certainly as the loss of our uric acid, the loss of our urate oxidase predisposes us to gout. So here again we have cogent evidence of a unique mutation which has been inherited by 4 species and which sometimes can be excruciatingly unfortunate. Maybe there's a secret, maybe there's a clue if we go a little bit further back in our history. We have another defunct gene in our DNA called the L-gluconogamalactone oxidase gene, which we can call GULO for short. And because GULO is deficient in our system, that is the basis of this money-spinning business of buying ascorbic acid, vitamin C in the dairies. We don't have the enzyme to make vitamin C, and neither do most of the primates. But the relics of that enzyme are still there in our DNA. Our DNA is a story of what was once functional, making proteins. And here is just a portion of that GULO gene present in our DNA. And you can see that there is a deletion, one of a number of mutations in fact, in humans, chimps, orangutans and macaques. Guinea pigs and rats don't have this mutation. In fact, guinea pigs have a mutation too, which is totally different, but rats have the functional gene. And this unique mutation again implies that it arose in a common ancestor of humans, chimps, orangs and macaques. The only implication of this is that when our vitamin C gene was lost, a long time back in our history, in our biological story, that imposed oxidative stress on us. And in fact, the main protection against oxidative stress in our blood now is uric acid. So it's possible that the loss of one gene promoted conditions which gave rise to the loss of another gene in our evolutionary history. I've got too much material here, so I'm just going to leave a lot of stuff out. But I'll just finish with one more, a pseudogene which is there in our history book. Our DNA is a history book. And this is a fragment of the vitellogenin gene. Now, vitellogenin is egg yolk protein. And the vitellogenin gene, or one of them, was last active in mammalian history in the platypus, which has one vitellogenin gene and which lays leathery little eggs. So this gene, in other words, is a survival of egg-laying precursors, of egg-laying ancestors. So the story in our gene, which establishes our biological origins, in fact, is a story which goes back beyond the dawn of the mammals. I must hurry through this next one because I do want to finish with some theological implications. Let's think of viral invaders. This is a picture of a cell, false colour of course, with viruses, green in this picture, budding off the surface. And this is just the summary of the reproductive strategies of this kind of virus. These are retroviruses which cause AIDS, and a number of types of tumours. And what happens when the virus comes along? It has an RNA, a little genetic code which is green, RNA which it injects into cells, and then that gets converted into a double-stranded DNA, and that double-stranded DNA becomes snipped into the genome of the host cell, which is a very clever thing to do if you're a pathogen. You just put your genome, your little genetic book, into the genetic book of the invaded cell. And now every time that cell divides, the viral genome just gets carried on with it. And here's just an example. The yellow represents a virus insert, and the blue represents cellular DNA on either side. Now the virus does something very cunning here. When it inserts, as part of its mechanism, it duplicates a little bit of the original cellular DNA. So here is a stretch of the original cellular DNA, and here's a GTTGC sequence, GTTGC there, duplicated GTTGC there. So this is called a target site duplication. It's a fingerprint of the way that viruses insert into DNA. Now we've got retrovirus genomes in our DNA, 400,000 of them, and they constitute 8% of our DNA. So it doesn't matter if you're from Africa, from the UK, from China, from the Middle East, we all have those same bits of DNA from retroviruses, 400,000 of them in our DNA. And the question arises, where did they come from? When were they added? Here is an endogenous retrovirus. Endogenous meaning they didn't get there through infection, they just got there through transmission from our parents as far back as we can see. And you can see what also looks very much like a target site duplication. TGA-GAC, TGA-GAC on either side. Well, that one is there in every human being. What about if you tried the chimps? And if you take chimp DNA and look at the same part of their genome, you'll find the same endogenous retrovirus with the same target site duplication, which indicates that the retroviral insertion was a unique event which occurred only once in a single cell, which has since been inherited by all humans and all chimps, and in fact, all gorillas. And what happens if you go further back? You get to the orangutan, and you find the orangutan has an undisturbed target site. That is the original target site, still preserved. There is no target site duplication and no endogenous retrovirus. And so you can just map many retroviruses onto a primate family tree. And I guess this grieves me, because now there is so much anti-evolution fervour amongst Christians, and yet amongst biomedical scientists, among geneticists, common descent is so firmly established that those religious people who gainsay it are only bringing discredit on the faith which they seek so ardently to proclaim. But we must pass on. I'll just tell one more story and then we'll reflect on some of the implications of this. This is one particular endogenous retrovirus called the ERV, endogenous retrovirus, WE1. And you can see it's there, ERV1, with its little target site duplication of 4 base pairs in humans, chimps, gorillas, orangutans, and gibbons, and also in Old World monkeys, although that's not shown here. But the original undisturbed target site is present in New World monkeys, prosimians, and in fact in dog, a very distantly related mammal indeed. Now this is of interest because when an endogenous retrovirus infects or comes to reside in DNA, because it's not part of our body plan, it just tends to mutate away. It doesn't do anything for us. And so it just accumulates mutations, there's no pressure to keep it active, and it just disappears into the genetic background. But this endogenous retrovirus, which has been there in primate DNA for something like 25 million years, retains an active gene. Its gene is called the ENV gene, envelope gene, which helps the virus to stick to cells which it's engaging. And that envelope gene is still intact. In 25 million years, there are no mutations which have occurred to inactivate it. And it makes a protein. And the protein, which is indicated here as squiggles and lines, that's its structure, is superimposable upon currently existing envelope proteins made by a mouse virus, Maloney-Murine leukemia virus, and in fact the yellow one, which is human T-cell leukemia virus, still causing cancer in people today. So here is an authentic viral protein still made in our DNA from an event which occurred 25 million years ago. What does this protein do? If you stain for the protein using antibodies, you can find that this viral protein is made very selectively. In fact, it's made in the placenta. And this just represents on the left-hand side some of the villi, the delicate fingers of the placenta which enable the baby's blood system to interface with the mother's. And if you stain for the viral protein, which is now called syncytin, you find that it's present in the outside surface of these little bits of tissue, these little placental villi, in a cell layer called the syncytiotrophoblast. And the syncytiotrophoblast is a fascinating cellular layer. It arises because cells fuse with each other. So individual cells start fusing, joining together to form giant cells filled with a nuclei from many cells. So the question is, does the viral ENV protein help in forming that syncytiotrophoblast? Does it make cells fuse together to induce the multi-cell structure? And here is an experiment done in culture. These are cells grown without syncytin, the protein made by the ancient ENV gene. And they're just growing as each of those little pink blobs is a stained cell growing on a plastic dish. On the right-hand side, you see cells in which syncytin has been expressed. And you can see a number of giant cells which are now fused, enormous cytoplasms, and they can contain something like 50 different nuclei. So the implication is that now an essential protein required for the development of every one of us was in fact introduced by a viral insertion event, what a geneticist would call a random event, because there's nothing planned about viral invasions. They cause AIDS and leukemia. And yet this mutational event has been selected to provide a protein upon which our lives now depend. Now, close your eyes. I'm going to move fast. So what does this mean? There is just so much evidence in our DNA for mutational events which now contribute novel and essential genetic structures. So our DNA is not only a history book which tells where biologically we came from. Genes describe genealogy. But our historical record tells how numerous genes have formed and developed and acquired new structures. What we need to do as we look at our genetic story is interpret it. We need to interpret our genetic history. I'd like to quote from George Knight, who is a New Zealand theologian, who speaks of the unique feature of biblical religion. And he says, Israel's faith was not the creation of a human mind, but a corporate interpretation of historical fact. Israel's scriptures were not essentially philosophy. They were not essentially ethics. They were not essentially law, although of course they contained that. Israel's faith arose from a corporate, multi-year interpretation of her history. This interpretation alone is based on fact. The prophets of Israel looked back at empirical history, and this comes back to what Jeff was saying, how Christian faith is a historical faith that is based in the empirical world. It's so strange, so utterly different from the thoughts of other human beings at any time in history, that the reader is compelled to ask whether it does not actually come from the mind of God. So as we look at the history in our DNA, which I believe is authoritative, it's God-breathed, because we are God's creation, we need to interpret it. And a theologian of 100 years ago, James Everard, said evolution reflected the orderly succession of causes which work out the rational purposes of God. This is not a religious alternative to a scientific conception of the world, but an interpretation of scientific findings from a religious point of view. So I believe as we look at the history in our genes, it must be interpreted from whatever religious tradition we come from, but in my case, from a biblical religious tradition. And here I move into an area where I am no expert. I was speaking to a historian just before, so I hope he will be sympathetic to me if I am very naive. But I recently read a classic by Herbert Butterfield, which is a description of an approach, a Christian approach to history. And Butterfield characterized human history in terms of a number of things. These leapt out. He didn't list them quite like this, but they leapt out to me as someone who thinks about our evolution. Butterfield characterized human history. He was a modern historian from Cambridge a couple of generations ago, celebrating Cambridge today, as showing natural process. To Butterfield, history showed natural process. It showed chanciness. It showed patterns and structure. It showed catastrophe and tragedy. And it showed progress. Butterfield was a great churchman. And as he looked at this history, he said there is nothing perhaps immediately obvious that history has the imprint of God's guidance in it. But he said from the perspective of faith, we can certainly see it. And so I'd just like briefly to work through these 5 headings. And there's not much to say about natural process. Certainly what I have described in our genes represents natural process. There is no sort of extra ingredient of genes suddenly appearing from nowhere. They come from duplications. They come from retroviruses. They are modified by jumping genes. So there appears to be natural process. And people say to me, well, where does that leave room for God in our creation? But exactly the same question can be directed to a historian who sees God in human history, God's hand in human history. where is there room for God in the history of the Tudors or the Majapahits or any other human empire? History seems to unfold through demographic change, and it's just the processes and flows of human activities. And yet God works in and through that. One of my heroes is Donald Mackay, and I know I've heard Professor Sieber speak of Donald Mackay in previous years in the university. Donald Mackay says, "I find nothing in what the Bible says about the dependence of our world on God to suggest that this dependence should be demonstrated by some scientific peculiarity of its past or present behaviour. Any view that God's being active in our world means that there must be something science can't explain is a complete non sequitur." That's so important. And I was so blessed as a young student to read Donald Mackay as one of the first Christians who described the interaction between science and faith, because it so clarifies things. Any idea that God's being active in our world means that there must be something science can't explain in physics or biology or chemistry or whatever is a complete non sequitur. Mind you, there is an enormous amount that science can't explain. Of course there is, and there always will be. But God is not to be found in that ignorance. The laws of nature we discover are not alternatives to divine activity, but simply our codification of God's activity in its normal manifestations. Well, secondly, we come to the category of chanciness, and I very much take on board what Bob said about speaking of natural evil. Clearly, there is no moral element in this. But we, as Christians, this problem of theodicy does rise. There is a chanciness to life. It's certainly there in our genes. The very things that have made bodily structures are also arise from processes which can cause disease. And here I do quote from Professor Polkinghorne, who says, as we invoke the free will defense to hold to faith in an active God in the affairs of history, so perhaps we do need a free process defense to keep our faith in the active of God in a world where there is suffering and sickness and where nasty bugs can also evolve. So Polkinghorne writes, free process defense, a free world with a capacity for disease and disaster, is superior to a wholly deterministic one. And this doesn't challenge our confidence in the sovereignty of God. Bob is so right in stressing that for Christians. But it does wonder, it points perhaps to the fact that God has withdrawn his sovereignty to give space for an authentic history. God is still sovereign, but allows history to follow its track, just as he allows us, as we see in the free will defense. A world in which people have the capacity to act in evil ways is better than a world of automata. And I see that freedom in me. God is sovereign. I am sinful. And yet God can still work out his creative purposes in my life, which is a tremendous comfort. And it's compatible with what we see in evolution. Evolution perhaps can disturb us because of what evolves. But within that randomness, God is working in and through it to bring history to its conclusion. There is patterns and structure in history. And perhaps we can come back to Menard and talk about chance and necessity. There is a necessity about the way biology is constituted. And I don't want to say too much because I think my namesake Graeme will follow up with this in more detail. Certainly Northcott, talking about human history, says for the ancient Hebrews, justice was not just a human invention, but a divine attribute set into the character and structure of creation. Even in human history, there is structure. It is not just random wars and human evil but there is set within the activities of what people do, the structure which perhaps is a pointer towards the God of justice from whom it flows. Let me just give you an example, again from the world of biology. Here is a small family tree of the mammals, and this is us up here. And this is the insertion event which gave rise to our syncytin gene. Now, placentae are very different placentas in different primate species. And mice also have an endogenous retrovirus which contributes towards the placenta of mice. And that's a different retrovirus. This is the mouse retrovirus. It's inserted into the DNA in a different place. It's a totally independent event. But a random event has produced the same outcome. And if you look at sheep, which are much more distantly related, they have a jaarziekte, excuse my old Dutch pronunciation. They've got an endogenous retrovirus of their own, which in this case is fascinating because it's still infectious. But this has also contributed a syncytin gene towards the sheep placenta. And you can do experiments on sheep, which you can't do on people. You can suppress the expression of that gene, and if you do it, the lamb aborts. So here are three random events. An endogenous retrovirus insertion. But there is a structure about it. There is a repeatability about it. Random events have produced the same outcome on repeated occasions. And here's another situation where a gene duplication, this gene up here, UTP14, which occurred early in mammalian, in primate history, has occurred also independently in lemurs, rodents, cows and elephants. So again, there is a structure in biology. Biology is not random. It is structured. There is an underlying fecundity to the way biology is structured. I quote here one of my favourite theologians who looks at history, and he says, Israel's history is the result of what both God and Israel do. For example, the particular course of the journey through the wilderness is determined by the interplay between God and the people. God who leads and cares, the people who complain and rebel. Polkinghorne speaks in biology and science of the interplay of chance and necessity. The theologian speaks of the interplay of human infidelity and God's faithfulness. And he says, in actual fact, history is formed by a faithful God and an unfaithful nation. And perhaps we can just move this sideways into biology and say that biology has arisen from the interplay between the freedom given to biological structures and processes and creatures and a holy, faithful and rational God who is involved in his creation at all times. Well, just briefly, there's catastrophe and tragedy. And people have said to me, an editor of The Listener once said to me when I was trying unsuccessfully to get something published, why should God use such an insanely slow and tortuous means of making life? This is what Bob has been talking about, and I don't need to add to it. And there's an artist's impression of an asteroid going into the Earth. But this is no new problem for Christians, because the Old Testament describes God as being the creator of ancient Israel. But look at the suffering and catastrophe of that history. And this is just a picture of the Romans whipping off the treasures from the temple in Jerusalem. And so, like it or not, the catastrophe in biology and earth history and the catastrophe in human history, which is given as God's creation. God is the creator of Israel's history, of ancient Israel's history. We must still understand that it is the same process at work. And so we arrive at the process, at the final step of progress. Is there progress in this history? Biological history and indeed human history. And I guess we intuit it. You start off maybe with matter, and then you find the wonder of life, which I really can only marvel at. Then you find cells with nuclei. Then you find multinucleated cells. Then you find animals with intelligence and consciousness, and animals with self-consciousness. Is there progress in this story? Some people interpret it differently. And our friend Professor Richard Dawkins says evolution has reached many millions of interamends, the number of surviving species at the time of observation. And there is no reason other than vanity to designate anyone as more privileged or climactic than any other. The historian must be aware of stringing together a narrative that seems, even to the smallest degree, to be homing in on a human climax. Here is a narrative with no end and no meaning, but it does not derive from science. It is purely an idiosyncratic interpretation of science. And I have there some things I just flogged from the internet: a fossil of a worm, and a fossil of a trilobite, and a fossil of a gastropod, and a fossil of J.S. Bach. And really, there is no ultimate distinction between them. But interpreting history, again we come back to these two books. To me, as a Christian, the hermeneutic key to understanding history comes from Old Testament history. In all its chaos, in all its suffering, in all its catastrophe and disaster. Because out of this history arose one who is the perfect representative of humanity. To me, the advent of Jesus Christ gives history its meaning. Out of this history, we find one who is the express image of God. And that enables me to apply these glasses to evolutionary history, because out of the mess of evolutionary history, its extinctions and its suffering and its pathogens and every else nasty thing which has evolved, out of that history arose mankind, which the Bible describes as humanity in the image of God. Yes, a provisional image. The Bible doesn't have provisional, but we must understand from Christ that humanity is provisional. And therefore we can look at evolutionary history and marvel at it, that it has produced, as God has worked in and through it, it has produced the creature that uniquely bears his image. And that gives me the confidence then to look forward through New Testament history. And one grieves at the mess of New Testament history, the follies of the church, the way the church time and again has fallen away from Jesus Christ, the terrible things done in the name of religion and the name of God. And yet see in that catastrophe, God is still working in his church, and he will bring it to its conclusion, to its denouement, to its climax. There is a climax in which the perfect image of God, made known, revealed in Christ through his death and bodily resurrection, will be conferred on creatures like us. And I find also great comfort in a creature like me, knowing that there is accident and chaos in my life, yet this same God is able to bring it to the end which he has determined. Thank you.
Thank you, Graeme. Well, time for a few questions.
Graeme, could I just check out before I ask the question that I've understood you correctly? You showed one sequence that led to human cancer, which had its origins in earlier primates. Am I correct?
Not directly, no. In fact, I did speed over it at one stage, but no, none of the changes I showed directly are related to human cancers. It is the processes which are responsible for human cancers, such as there is a retrovirus in the world now called human T-cell leukemia virus type 1, which inserts into DNA just like those ancient ERVs did, endogenous retroviruses did, but which cause a significant burden of human disease at present. So they are potentially pathogenic viruses, but they are also potentially capable of producing new structures which are now essential for our survival.
So, if I understand you correctly, then that there are in our genetic code the potential common not just to humans, which may result in human cancer, which may result in human cancers. Other factors obviously coming into play as well as the genetic code.
Yes, again, I would say the processes. My mind's gone a little bit blank because you're probably right, but I just can't quite think of a concrete example of an event in evolution which is also integrally involved in a particular disease. I guess the uric acid one is probably an example of an event which presumably was of benefit, but which now causes gout in some people.
Because I would relate that back to the discussion I had with Bob White about natural evil.
Yes.
Because that would seem to me to be a case of the potential for suffering and pain that may not be attributed to human fault or carelessness, that it's part of our genetic code that there is pain and suffering, and if that is the case, then there is a class of natural evil to which you cannot invoke St. Augustine's free will defence.
Except if you use John Polkingham's argument that there is a free process defence. This is not a clockwork universe. It has an authentic history of its own. God upholds everything that exists, but clearly he does not cause everything that exists. I don't know whether you're happy with that, Bob. God upholds everything that exists, but he has given freedom to its workings so that what happens is not directly caused by God. I mean, the accident down the road which kills a child is not caused by God. Rather, God grieves over it. God is a grieving God. He grieves over it, but he does not cause it.
Yes, I was challenging Bob. You may want to come in on this. Bob said there's no such thing as natural evil. I was challenging that, but it seems to me that there are some examples from your genetic stuff which would also be attributed to natural evil.
Again, is it the use of language which is the problem? That evil, if we speak of evil, we should really use it for moral, for events which have a moral underpinning.
Well, in the debate in the problem of evil, the key arguments come from St. Augustine evoking the free will defence. But in the debate about that, natural evil cannot use that defence because it's not attributed to human causes. It comes from natural events, right? And it seems to me that you were giving some examples of natural events that cause pain and suffering. So therefore you can't evoke the free will defence.
Not the free will defence. I mean, what I did hurry over this, we have jumping genes in our genome. In fact, invaders to our genome occupy nearly 50%. We've got 2 meters of DNA in every cell. Half of that has been imported from jumping genes and viruses. And they jump in a way which is fairly individualistic. And yet they have contributed to our biology in a way which is only now just starting to be appreciated. So it's incredibly likely, it's almost certain that they have contributed to our appearance on the earth as human beings. But they certainly also do engage all the time in mutational events, which are the source of all kinds of diseases. In fact, I slipped over cystic fibrosis as being a tragic outcome of the biology of these jumping genes, which have nevertheless colluded to make us human beings.
Well, perhaps we should move on from that. I think there are some interesting issues, although I think this widening out from free will to free process also probably responds to your concern, but maybe we can talk about that later. Any other issues?
Yes, Graeme, I'm interested in your opinion. If we were very lucky enough to find some DNA from Jesus Christ, right, and we analyze it, would that have traces of Joseph's DNA or genetics? Or how would it look like?
That's right. According to the Bible story, the advent of Christ was a break from normal expectation. And so as a Christian, I accept what Matthew and Luke tell us, and what certainly Paul implies, that the conception of Jesus Christ was a work outside what we normally understand by science. So no, in that hypothetical situation, I don't believe that there would be evidence of of the Y chromosome of Joseph. But of course, you're referring here to what we would all regard as a total mystery. It is a mystery. Just as I think it was Jeff was speaking, I also thought of a recent article in Nature, an essay by a sociologist of science who is calling for rapprochement between sociologists and science. There's been so much bad feeling for years. And he says that scientists have to be more humble. He says scientists have to realize that there are a lot of things that they cannot speak authoritatively on, and that science cannot tell what will happen tomorrow. So the laws of nature are all provisional. But the Christian believes that God is the source of those laws, and that they will have different manifestations as God works under different conditions.